Everybody, Dr. Becker. We are going to jump into our first big lecture here, which is talking about chemical and biochemical foundations. And this is going to be divided into two PowerPoints here. So this is part one. We're going to jump back in time and start reviewing again chemistry and biochemistry and some of those other foundational concepts that we learned in undergraduate. If you're like me, we learned them a long time ago, and that's okay. So everybody's going to be at a different spot of how fresh this content is in your mind. If you need to take your time and to come back and forth and jump in back and forth, that's fine. Just take your time and review as you can. We're going to go back to talking about atoms and ions and covalent bonds and extracellular fluid, intracellular fluid, the periodic table of elements, etc. What we're kind of doing is doing kind of a quick highlight of a variety of different topics. We're not going greatly in depth, but what we are going to do is review the major topics that this text feels we ought to have fresh in our mind so that we can move into the other chapters. Chapter 2, 3, and 4 to me is very boring because this is not the kind of stuff that I enjoy. This is cells and atoms and bonds and the rules of chemistry and the rules of cellular working and genetics and all of those things. So if you are like me and you need to take your time and come back to it, you're certainly welcome to do so. So pause as needed. This one here talks about some of the key concepts that we are going to be working through. All right, so one of the first things to talk about is the chemical foundations of life. Remember that matter is anything that has mass and occupies space. So liquids, solids, and gases are all of the types of matter, and the atoms are the building blocks. So an atom is a substance that consists of, an element is a substance that consists of only one type of atom. We have 118 elements that are recognized, but our bodies are mainly made up of what we call the big four: oxygen, carbon, hydrogen, and nitrogen. Okay, so if someone said, what are the big four elements in our body? Oxygen, carbon, hydrogen, nitrogen. We have 20 other plus elements that are considered to be lesser, and those are these listed here, like calcium, sulfur, etc. Each element is characterized by unique physical, chemical, and biological properties, and we're not going to get stuck on those, but just again, a very brief overview. We have the big four main types of atoms and elements in our body, and then we have a bunch of lesser elements as well. We have to give a good review of the atomic structure here, okay? So the atom is structurally divided into two regions. We have the nucleus here and here, and the nucleus is made up of protons, which are positive, and neutrons, which are zero. The electrons then are what orbit around the nucleus. So protons are positive, and neutrons are neutral. They don't have a charge. And then the electrons are negative. Taken together, these particles give the nucleus a net positive charge. Remember, because if you had two protons and two neutrons, the neutrons are neutral, so you have two positives. So this is now going to be a positive charge, and the electrons float around in the electron cloud in these rings, and those will have a negative charge. Because the electrons are negative, they are attracted to the nucleus and will naturally orbit around it. Atoms are electrically neutral because the number of protons equals the number of electrons. So this is 0, 0, positive, positive, negative, negative. So it all equals itself out and they're neutral. Atoms then here are considered to be neutral. When we talk about the periodic table of elements, we're not going to have to memorize any of things here, but let's kind of review the general parts of the periodic table of element and the concepts around it. The elements are classified according to their atomic structure. So in the table, each element has a specific symbol, like C is for carbon. The atomic number is the number of protons in the nucleus, and it's used to arrange the elements. So we start at the top left corner of the table and continues in a series of horizontal rows. Hydrogen, which only contains one proton, and so on, and we move across, right? A period is the name given to the horizontal row of elements in the periodic table. And this arrangement also places elements in 18 vertical groups as well. So hydrogen, lithium, sodium, potassium, etc. are all belong to the first group here. Okay, so horizontally we have the periods, right? Okay. The reason that this is helpful is because it's going to help us understand how these different elements would react with each other. So let's review that concept here. The electron shells, right, are that outer layer. So again, the yellow is the nucleus, and then the electrons form a ring around it, and you can have multiple different rings on the element. So the electrons then are arranged on different energy levels, and we refer to these as shells. So instead of calling it a ring, it's a shell. So hydrogen only has one shell, carbon has two shells, potassium has 1, 2, 3, 4 shells. Okay. The higher energy levels are farther from the nucleus and are only occupied with electrons if the inner shells are already full. So let's talk about that. Because electrons have a negative charge, they have a tendency to repel each other. So it's that repulsion that determines the number of electrons that can occupy each orbit. Remember, the first electron shell is the smallest and only allows up to 2. So we have some rules here that we need to know about. So the first electron shell is the smallest and it only allows 2. But the higher shells found in the elements with higher atomic numbers allow up to 8 electrons. We call this the octet rule. Okay. So the electrons found in the outermost shell is referred to as the valence electrons, and we call this shell the valence shell. So it's the electrons found in the valence shell, the valence electrons, that determine the reactivity of an atom, meaning how can they interact with other atoms to form chemical bonds. So we're going to talk a little bit about that again. But first, when we talk about the octet rule, what does that mean? So it helps determine the reactivity of the element. Okay, so the octet rule states that atoms will strive to occupy their valence shell by ultimately collecting 8 electrons in that energy level. So elements with 8 valence electrons, such as argon, are very stable, and they actually don't want to interact with many other atoms because they're already full. So their valence shell in argon already has 8. So it's kind of like the bus is driving by and all the seats on the bus are full, so they just drive by. It doesn't matter who's standing on the side of the curb shouting for their attention. They're like, nope, we already have 8. We're cruising right by. We're very stable and predictable. When you have an atom or an element that doesn't have 8 in its valence shell, it's considered to be reactive, meaning if that bus is driving by and there's only 7 passengers on it and they see people getting rowdy on the street corner, they're going to stop and interact and react with them and try to pick up a passenger because they only have 7. So we consider those elements very reactive. They like to associate with others. They're going to stop and talk to people on the road, except for hydrogen, helium, and lithium. Okay, so when this says incomplete filling, that means they don't have 8, but there's 3 here that are a little different. Hydrogen is only 1 electron, and it's a good chemical reactivity. Okay, but helium has 2 electrons, that fill the outer shell, and it's actually considered chemically inert because remember, the first shell should only have 2. It's after the first shell that needs 8. Okay, so in the first shell for helium, it is incomplete because it should have 2. So helium is very active. It is very social. When it's a bus driving down the road, the windows are all down, and this one electron is going, hey guys, I have a seat on the bus over here. Come hop on with me. Okay. Helium is the exception that it has 2 already, but it only has 1 shell. So it is considered full and it's chemically inert. It's not going to pick up passengers. Okay. But something like carbon, okay, the first shell is full with 2. So after the first shell, the octet rule applies. So its valence shell only has 4. So it is reactive. It's going to pick up other passengers and it's going to play well with others because they have 4 seats on the bus. I hope that analogy makes sense. So when an atom does not have the full valence shell, it wants to fulfill the octet rule, and they're very reactive. Now, it doesn't mean always that they have to fill their seats. So potassium only has 1 out here. So they want to fulfill the rule. Well, there's multiple ways that this potassium can be filled. I can either pick up 7, and now I've satisfied the octet rule, or I can get rid of 1, drop a ring, and now I'm fulfilling the octet rule because my ring has 8 underneath of it. So when we talk about the octet rule, remember, it's not always picking up passengers. It's also maybe you just get rid of a passenger and now you've gotten rid of a ring and they're just as happy. Hope that makes a little bit of sense for y'all. So let's go into a couple of different examples here. So when you gain or lose an electron, we are creating ions. So let's talk about here sodium. So we have Na. You can see on its most outer ring it only has 1. So its valence shell has only 1 valence electron. It has the number 11 because ultimately it has 11 protons and 11 electrons. Okay, so it has the 3 shells. The first shell has 2, so that's satisfied. The next shell has 8, so that's satisfied. The last shell only has 1. So in order for sodium as an atom to fulfill the octet rule, it is easier actually to lose that electron than try to gain 7. So one of the very common things sodium wants to form an ion with is chlorine. So we have sodium chloride. We talk about that as an electrolyte in our body all the time, sodium and chloride. And an ion is sodium chloride. They're together. So what happens is we're going to lose this one here, give it to chlorine. So chlorine had 7, now it's happy because it'll have 8. And now they're bonded together. And it has an ionic bond between sodium positive and chloride negative. So when sodium released that 1 electron, it completely filled its middle shell, right? Because it lost its outer shell, and the atom achieved stability, but loses its neutrality, and it's now positively charged. So now we got rid of 1. So remember, here is balanced because it had 11 out here and 11 in here. But now it got rid of 1, so now it becomes positively charged. So chlorine only needs the 1 electron to fill the octet rule. It is a good electron acceptor, and it can become negatively charged. So if an atom of sodium donates an electron to the atom of chlorine, both atoms fulfill the octet rule, right? And they're considered stable. But after the electron transfer, sodium is left with 11 protons and 10 electrons, resulting in a net positive charge. Similarly, once chlorine acquires the extra electron, now it has a negative charge. So atoms that have an overall charge are called ions. Negatively charged atoms are called anions. Positively charged atoms are called cations. And then remember, the ionic bond is a chemical bond that is formed when one atom donates 1 or 2 electrons to the other atom. And that transfer leads to the formation of a cation, right? Okay, so sodium and chloride, as well as several other ions, obviously are very critical in their function in our body. The next topic the book kind of jumps into is the concept of water molecules and what happens in water when you have ions that come into a water solution. So water is about 60 to 70% of our body weight, and most ionic compounds in the body are found dissolved in water, which we can see here. The polarity of the water is something we're going to talk about later down the road. This picture here is just the organization of water molecules. So it's H2O right here, right? Now when you have sodium chloride as an ion come in, they're going to dissociate chloride and sodium. The ionic bond is broken, and then the hydrogen atoms are attracted to the chloride, and the oxygen atoms are attracted to the sodium. The ions of the body are all stable when surrounded by water molecules. Because now that this ionic bond is split, these now go back to being neutral, and water is also considered neutral. So when you have ions that break apart into water solution, now the water molecules and all the molecules are stable. So as they're dissolutioned into the water, they become stable again. Acid and bases also will dissociate in the solution of water. Acids are proton H-positive donors. Bases are proton acceptors. So this example is hydrochloric acid, HCl. It's a very strong acid, and acids are donors. So when it dissolves in water, that ionic bond is broken. And again, the hydrogen atoms of water are attracted to the chloride, and then the oxygen atoms of the water add the H plus and become hydronium ions. I don't expect you to memorize these specific things. This is just illustrations to illustrate the point that we need to understand. When we have sodium hydroxide, which is a very strong base, and remember bases are proton acceptors, it will dissolve in the water, and the sodium then becomes surrounded by water molecules, and the hydroxide ion is stable. Okay, so when we're talking about the water and how the ions act in water, we have to review a couple of different concepts of intracellular and extracellular fluid, because the body fluids differ in composition depending on whether they're intracellular or extracellular. Okay. So I'm going to pause the video here. I'm going to turn on the osmosis video and we'll listen to it together, and I'll come back to the lecture.
The key to life, and it takes up a big proportion of our body weight, typically around 60%. The precise amount of water depends on a person's body composition. Since fat doesn't store any water, a person's water content is inversely proportional to a person's fat content. So a really muscular and lean person would have a relatively high proportion of their body weight made up of water. Additionally, females tend to have more fat than males, and so on average tend to have a lower proportion of their body weight made up of water. Total body water can be divided into 2 major compartments: intracellular fluid, which is fluid inside cells, and extracellular fluid, which is fluid outside the cells, like in the blood and in the interstitial tissue between cells. Assuming that the total body water is about 60% of a person's body weight, roughly 2/3 of that, or 40%, is intracellular fluid, and the other 1/3, or 20%, is extracellular fluid. And this is known as the 60-40-20 rule. Intracellular fluid is important for dissolving cations, which are molecules with a positive charge, as well as anions, which are molecules with a negative charge. The major intracellular cations are potassium and magnesium, whereas the major anions are proteins and organic phosphates like ATP. Fluid compartments always maintain the same concentration of positive charges as negative ones, in order to stay electrically neutral. and this is called the principle of macroscopic electroneutrality. So, for example, the potassium in the intracellular fluid is balanced out by negatively charged proteins and organic phosphates. Interstitial fluid, which is the fluid that can be found surrounding the cell, and plasma, which is the aqueous portion of blood. The major cation in extracellular fluid, both in the interstitial fluid and in the plasma, is sodium, and the major anions are chloride and bicarbonate.
To be tested on the rule of neutrality, by the way. Gotta go back a
Cellular
little
fluid
bit.
can be subdivided further into interstitial fluid, which is the fluid that can be found surrounding the cell, and plasma, which is the aqueous portion of blood. The major cation in extracellular fluid, both in the interstitial fluid and in the plasma, is sodium, and the major anions are chloride and bicarbonate. Now, the plasma makes up about 55% of the blood, while the remaining 45% is mostly made up of red blood cells, white blood cells, and platelets. Plasma is made up of around 90% water, and 10% proteins like albumin, which helps transport hormones and minerals. Interstitial fluid and plasma are really similar, and that's not surprising since plasma leaks out of the blood and goes into the interstitium through tiny pores between endothelial cells in the capillaries. These tiny pores between capillaries let small solutes in water pass through but block large proteins in cells. In fact, because some proteins are too large to pass into the interstitium, a phenomenon called the Gibbs-Donnan effect helps the plasma remain electrically neutral.
You are not going to be tested on the Gibbs-Donnan effect.
The Gibbs-Donnan effect is when the abundance of negatively charged large proteins repels smaller anions into the interstitium while attracting small cations into the plasma. So compared to the plasma, interstitial fluid tends to have higher concentrations of small anions like chloride and lower concentrations of small cations like sodium. Ultimately, though, the difference between plasma and interstitial fluid is small, but the difference between intracellular fluid and extracellular fluid is pretty large, and this large difference is physiologically important. For example, sodium-potassium ATPases help to establish a high concentration of potassium inside the cell and a high concentration of sodium outside the cell. Establishing these differences between intracellular and extracellular environments is incredibly important for allowing cells like neurons to fire action potentials, among many other functions. Now, measuring the volume of each of the body fluid compartments can be done using what's called the dilution method. That's where various substances which settle in a specific fluid compartment are given to an individual and their concentration is then measured. For plasma, it's done with radio-labeled albumin, because albumin is a large protein that cannot move into the interstitial space. For the interstitial compartment, it's done with slightly smaller molecules like mannitol and inulin, which can pass through the capillaries into the interstitium but cannot cross the cell membrane.
You're not going to be tested on that either. I'm going to fast forward a little bit here.
So, by injecting a known amount of a substance and allowing the substance to diffuse through the fluid compartments, it's possible to measure the concentration of that substance in the blood, as well as if any was lost in the urine. Ultimately, it's possible to calculate the volume of distribution via the equation volume equals amount divided by the concentration. Here's a sample problem. A 70-kilogram man is injected with 150 millicurie of D2O.
Not going to get that in depth. Skip that section here.
Alright, as a quick recap. The 60-40-20 rule states that the total body water takes up about 60% of our body weight. And more specifically, 40% is intracellular fluid and 20% is extracellular fluid. Measurements of these fluid compartments uses the dilution method, where various substances are injected and allowed to distribute differently among these fluid compartments. Final concentrations from the plasma are measured and used to calculate the final volume of the fluid compartments.
All right, that's a quick little review there. You're not going to be tested on the specifics in this video. It's just something that a good illustration can help reinforce the ideas we're talking about. So to recap, the intracellular fluid is the fluid within the actual cells. The interstitial fluid is actually part of the extracellular fluid between the cells. And then blood plasma is the second part of extracellular fluid. So to review again, extracellular fluid is rich in sodium chloride and bicarbonate. The plasma has those protein anions, and the interstitial fluid has few proteins. Intracellular fluid is rich in potassium, phosphorus, phosphate, and the proteins. Okay. Remember that when we're talking about ionic bonds, too, that we have these particles that do have electrical charges, and so it can conduct an electric current in the solution. All ions are electrolytes, so the ions in the body are abundant in the aqueous solutions found inside and outside the cells, and the presence of specific ion pumps and channels are present, and these ions are differentially distributed between the intracellular compartment and the extracellular compartment, which is the interstitial fluid and plasma. So sodium chloride and bicarbonate have higher concentrations in the extracellular space. Potassium, phosphate, and the protein anions are highly concentrated intracellularly. And this differential distribution of those ions also contributes to the generation of what we call our membrane potential. The cells that have an overall positive charge on the external side of cell membranes and a negative charge on the internal side. When we have excitable cells, so think about like our cardiac cells, our cardiomyocytes, alterations of these charges may cause an action potential. Same thing with our nerve cells and those things, right? The alterations of those charges is what can cause an action potential. Okay. Next review is about the acids and bases again. The acids and bases can form these ions in the aqueous solutions of our body. A definition of an acid depicts them as molecules that will release protons, which is H+, the hydrogen ions, when placed in an aqueous solution. So you're considered an acid when you are placed in water, essentially, you will release a proton. Acids are the givers, typically. The process of dissolving the acids into water molecules binding the protons will form hydronium ions, which is what we can see in that figure there. So remember, substances that release protons when placed in water, like carbonic acid and phosphoric acid, are considered acids because when the ion bond is broken in water, they will release the H+. They will give up that positive. So those molecules are acids, but they do not all behave exactly the same way. Acids are classified based on their tendency, remember, to give up that proton. They're a giver. But strong acids have a tendency to release protons that do. Strong acids have a greater tendency to be able to give up that proton than weak acids do. The example is hydrogen chloride is considered a strong acid as its ionization in the water is virtually complete, meaning that all molecules are separated into protons and anions. It's considered a strong acid because it can dissociate completely. But carbonic acid and phosphoric acid are only partially able to break apart in the water solutions, and they're considered weaker acids then. Once an acid is dissolved in the water, the ions formed then can conduct that electrical charge or the electricity and are therefore called electrolytes. So apart from the HCl formed in the process of like gastric acid secretions, most acids in the body are considered weak acids. Okay. So we're going to keep going with this concept. We're going to review a base. So the base is a compound that can dissociate, and it's a taker, and it releases hydroxide ion, which is the OH negative, and a cation. So the bases can accept a proton. They can become positively charged. Therefore, the bases are defined as proton acceptors. So if you had something that's a base, it's going to accept the proton. Dissociation of sodium from the hydroxide ion provides a free hydroxide that is capable of neutralizing acid. So let's take the example of bicarbonate ion, which is HCO3 negative. So it's a base that plays a major role in our ability to maintain our pH between 7.35 and 7.45. So bicarbonate ion, HCO3 negative, is very important in our homeostasis. One homeostatic role of the bicarb ion is that it is as it relates to GI function. So bicarbonate is produced by the pancreas and is released into the duodenum to neutralize the acidity of the chyme that comes from our stomach. As an example, pancreatic bicarbonate secretion is necessary because while the stomach is usually well protected from acid, the duodenum and small intestine is not. So acid-induced damage can result in gastric and duodenal irritation and ulcers. So just like acids, bases can be classified depending on their ability to dissociate in water, and physiological bases are generally relatively weak. Okay. So the bicarbonate carbonic acid buffer system is centered on carbons and bicarbs, right? So carbonic acid, when we put it into water, remember it's an ion, it's going to dissociate and it becomes water plus CO2. The CO2 can be removed by the lungs if we hyperventilate, or it dissociates into bicarb plus H. Either can be secreted or reabsorbed by the kidneys to maintain balance. So to review, what do we need to know? Strong acids can completely dissociate in water. Weaker acids don't tend to. An acid is a proton giver or hydrogen giver. Okay. They tend to want to give up that proton of hydrogen. The base is a compound that can dissociate but wants to release the hydroxide ion and a cation, but they will accept that proton of the hydrogen typically. Again, stronger bases can dissociate in water. Weaker bases cannot completely do that. Acid and bases are what we need in our body buffer system to maintain that pH. So when we are talking about the pH of the body, and we know the 7.35 to the 7.45, what we're really talking about is an expression of the concentration of the hydrogen ion in that solution. So as everything is mixed together in that fluid, the acids come in, they release the proton hydrogen, the bases come in and they want to pick up the hydrogen. So we want a balance of that process to be reflected as a pH between 7.35 and 7.45. So it's an algorithm type thing, and we are not going to even begin to explain how that works in this class, other than the basic concept of a solution or anything that is less than 7 of a pH is considered to be acidic, and above 7.35 is considered, or less than 7.35 is acidic, greater than 7.45 is basic or alkalotic. To maintain the acid-base homeostasis is paramount to how we function in the human body. Remember that we need our renal and our respiratory symptoms together with our chemical buffers. in order to keep this process running. The bicarbonic acid buffer system can accommodate more than 80% of the excess ions produced or lost by the body. Okay, that kind of wraps up that little component that they're talking about there. Now we're going to kind of switch to the next line of review and talk about covalent bonding. So the ionic bonds are not the only strategy that an atom can use to fulfill the octet rule. The other thing it can do is share rather than exchange that electron. So when electrons occupy a single orbit that's common to two atoms, they form a covalent bond. So that bond may allow both atoms to occupy their valence shells and have a stable electron configuration. So if you have one electron contributed from each, it's a single covalent bond. If you share two electrons from each, it's a double covalent bond, and 3 is a triple. So here are some examples of covalent bonds. Remember, we talked about carbon has 4 electrons in the valence shell and therefore needs 4 more to fulfill the octet rule. Hydrogen has only 1 electron and needs another one to be stable because it only has that 1 ring, so instead of 8 it only needs 2. So 1 atom of carbon can interact with 4 atoms of hydrogen, and each hydrogen atom shares 1 electron with the carbon atom, and that becomes carbon atom with 8 electrons in the valence shell and 4 hydrogen atoms with 2 electrons. And that bond formed between the carbon atoms, carbon atom 1, and each of the hydrogen atoms are called a single covalent bond because there's only the one here. And this is what we get that results in methane gas. And we would express that as that formula. So this one here is showing you a single carbon and the 4 hydrogen atoms will bond together. They make methane. The 2 atoms of oxygen can be joined by a double covalent bond here, forming molecular oxygen. And then here is 2 nitrogen atoms can be joined by a triple bond, forming molecular nitrogen here. So this is an example of some of those covalent bonds. Again, I'm going to pause the video here and listen to some extra information about covalent bonds from osmosis. Remember, osmosis is just a review, it's not testable material.
A covalent bond is where electrons are shared between 2 nonmetal atoms. Let's start with hydrogen, which is a nonmetal on the left side of the periodic table. Hydrogen has an atomic number equal to 1, which means that it has 1 proton and 1 electron. If we take 2 hydrogen atoms, each with its own electron, we can combine them to form a molecule, where each atom contributes 1 valence electron to the final molecule. In other words, these 2 valence electrons are shared by both atoms in the final molecule. We can represent this process by putting 2 dots between the 2 hydrogen atoms. These 2 dots represent 1 covalent bond. We can also represent a covalent bond by drawing a straight line between the hydrogen atoms. One straight line means 1 covalent bond and represents 2 valence electrons. Now, in our hydrogen molecule, opposite charges attract. So the 1 electron from the hydrogen atom on the left is attracted to the positively charged nucleus of the hydrogen atom on the right. At the same time, the negatively charged electron from the hydrogen on the right is attracted to the positively charged nucleus of the hydrogen on the left. So a mutual attractive force holds together the 2 atoms in the covalent bond. At the same time, like charges repel, which keeps the two positively charged nuclei from getting any closer together. Another way to represent the hydrogen molecule is by writing H with the subscript 2. This implies that we have two hydrogen atoms in our molecule. So hydrogen is an example of a diatomic molecule, which is a molecule composed of two atoms.
We tested on the term diatomic molecules in our book.
There's an easy way to remember a list of the elements that form diatomic molecules. Imagine a mad scientist whose name is Dr. Brinkelhoff, whose name spells out each of the diatomic molecules. Br is bromine, I is iodine, N is nitrogen, Cl is chlorine, H is hydrogen, O is oxygen, and F is fluorine.
This is not testable material, it's just a good review.
Let's try drawing some of the structures of these molecules, starting with the example of the fluorine molecule, F2. Fluorine is in group 7A on the periodic table. Because the group number is equal to the number of valence electrons, we know that each fluorine atom has 7 valence electrons around it. Fluorine is most stable when it has a full set of 8 electrons around it, which is referred to as an octet. For this reason, it will tend to form bonds that allow it to get an octet of electrons. It gains an electron by sharing one of the 7 electrons from another fluorine atom to create a covalent bond. There are therefore 2 shared electrons in the covalent bond, one from each atom. Each fluorine atom in a diatomic fluorine molecule has a full octet, consisting of 7 electrons from its original set, and the 8th electron shared with the other neighboring fluorine atom. Now let's look at the oxygen molecule, O2. Oxygen is in group 6A on the periodic table, so each oxygen atom has 6 valence electrons. We can draw these in around each oxygen atom. We can see that we still don't have an octet around each oxygen atom, because each oxygen atom is missing 2 electrons from its octet. So to get an octet of electrons, the atoms need to share 4 electrons with one another. And that happens by having a double bond between the 2 oxygen atoms. We indicate a double bond with a double line in drawings of the molecule. Let's look at the nitrogen molecule, N2. Nitrogen is in group 5A on the periodic table, so each nitrogen has 5 valence electrons. And we can draw these in around each nitrogen atom. And once again, we can see that we still don't have an octet around each nitrogen atom because each nitrogen atom is missing 3 electrons from its octet. As a result, 2 nitrogen atoms will share 6 electrons with each other, resulting in a triple bond between the nitrogen atoms and an octet around each nitrogen. You can probably see where this is going, but we indicate a triple bond with a triple line in drawings of the molecule. Molecules don't have to be diatomic. For example, we can draw out the dot structure for methane, which has a chemical formula CH4. Carbon is in group 4A on the periodic table, so carbon has 4 valence electrons. We can draw these in around the carbon atom. Next, recall that hydrogen is in group 1A, so each hydrogen has 1 valence electron. Notice how the 4 hydrogens all fit around the central carbon to form 4 carbon-hydrogen bonds. Carbon is now surrounded by 8 electrons, resulting in an octet, and thus our final stable molecule. You should notice that hydrogen does not follow the octet rule. That's because the first period has a shell that holds a maximum of 2 electrons. Since hydrogen is in the first period, it is stable with only 2 electrons around it. Finally, we can look at water, which has a molecular formula of H2O, which means that it has 2 hydrogen atoms and 1 oxygen atom. We already know that hydrogen is in group 1A and oxygen is in group 6A on the periodic table. So each oxygen has 6 valence electrons. We can draw these in around the oxygen atom. As before, each hydrogen has 1 valence electron. If we draw out the water molecule, then we can see how each hydrogen shares its electron with oxygen. The final dot structure shows that oxygen has an octet of electrons around it, and each hydrogen has 2. Alright, it's a quick recap. Covalent bonds form when nonmetal atoms share their valence electrons among themselves, in order to allow all the atoms to have a full octet. Hydrogen is one of the exceptions to the octet rule, because it ends up with only 2 electrons around it. Many elements, such as oxygen, nitrogen, and hydrogen, form diatomic molecules. In a water molecule, an oxygen atom gains an octet of electrons by sharing electrons with 2 hydrogen atoms.
That was a nice osmosis video to help us kind of get an illustration of the things we've talked about from another perspective. Next that we go into talking about is the bond properties in a little bit more detail here. So non-polar versus polar covalent bonds. Okay, so we're still talking about covalent bonds. Electronegativity is defined as the tendency of an element to attract a bonding pair of electrons. So electronegativity depends on the number of protons in the nucleus. So an element with more protons will be more electronegative than an element with fewer protons. Since elements that have the higher proton number also have higher group number, electronegativity increases across the period in the periodic table. For example, oxygen, which is 6A, is more electronegative than carbon, which is 4A. But atoms that belong to the first or second group, such as hydrogen or beryllium, have a greater tendency to release valence electrons and are less electronegative. So for that reason, they are sometimes referred to as being electropositive. Bear with me. When a covalent bond is formed between an atom that has that higher electronegativity and an atom that has lower electronegativity, electrons will spend more time in the vicinity of the electronegative atom. So let's say that one more time. When you have a bond, a covalent bond formed, an atom that has that higher electronegativity and the atom that has the lower electronegativity, the electrons are going to want to spend more time in the vicinity of that electronegative atom. So this in turn confers a partial negative charge around that electronegative atom and a partial positive charge around the other atom. And so the development of a positive pole and that negative pole is what we call a polar bond. And the polarity of that bond depends on the electronegativity difference between the 2 elements participating in the bond. Okay. So let's talk about that here. So as shown in this table here, 2 elements with a large electronegativity form a non-polar covalent bond, like oxygen. So when something, well, let's say this way, a polar covalent bonds have unequal electron sharing, meaning one atom has a stronger pull on the bonding electrons. Okay, so this results in molecules with partial charges shown as D plus or D minus. Okay, so if something is non-polar, the differences between the 2 atoms is very low. So oxygen, for example, is considered non-polar. But a polar covalent bond means that the difference between the electronegativity of the 2 atoms is a big enough difference that one's going to have a stronger pull on those electrons than the other. That is water. But an ionic bond is when the electronegativity difference is actually greater than 2, and that's sodium. An example is sodium chloride. So when we're talking about polar versus non-polar, important to understand, it's the difference of the electronegativity difference. If it's a polar covalent, that means that one atom has that stronger pull, that bonding electron that they share together is going to lean more towards that highly electronegative atom. Under the table of 2.1 in your book, it's a little paragraph there that I think you guys should read for sure and understand. The concept of the polar and non-polar covalent bonds later in this chapter becomes important because hexose sugars, which are rich in hydroxyl groups, have many polar regions because of the electropositive hydrogen atom bound to electronegative oxygen atoms. And that oxygen atoms will have a stronger pull, and you'll have a polar covalent bond. Nucleotide bases have polar regions due to electropositive nitrogen or oxygen atoms that are bound to neutral carbon or electronegative hydrogen atoms. And these elements contribute to hydrogen bonds holding together complementary base pairs in the double-stranded DNA. So sugars and nucleotides tend to be polar molecules with partial charges in the bonds connecting oxygen or nitrogen atoms to hydrogens. But fatty acids that make up our triglycerides and our phospholipids have long stretches consisting entirely of non-polar covalent carbon-hydrogen bonds, making those molecular regions nonpolar. And so the book goes into describing what they call molecular geometry. So the repulsive forces operating among these groups of electrons determine the molecular geometry. So consider a molecule that has 3 atoms connected by 2 bonding pairs, and we're looking at figure 2.9 in your book. Since the electrons that constitute bonding pairs are negatively charged, they will repel each other, creating a linear line. So figure 2.9a, that's a linear line there. But a common biological molecule that illustrates linear geometry is CO2. But in B, that's methane. Remember, that's the carbon with the 4 hydrogens, and they actually form a tetrahedral geometric shape. We're not going to have specific questions on that in here, but we have to understand the concepts here. So repulsive forces in molecules with 4 electron groups determine tetrahedral geometry. So that's a little bit of a blurb. If you're totally confused by that, don't worry. Don't freak out about it. Look at figure 2.9 and just realize that in the way that the atom is shaped, there is some different geometrical patterns that can result on the different bonds that occur. All right, moving on to the molecular properties of water. So in a water molecule, the bonds between the oxygen and the hydrogen atoms are polar. Okay, that means that there is a pull of these 2 atoms that come together that move their shared electron closer or pulled more towards one than the other. So electrostatic attraction forces will develop between the slightly negative charges on the oxygen and the slightly positive charges on the hydrogen in the neighboring molecules. But this is also could be referred to as a hydrogen bond. So each water molecule can make up to 4 hydrogen bonds with neighboring water molecules and is subject to pulling forces in different directions. On the surface of a liquid, the molecules can make fewer hydrogen bonds. Thus, each molecule is more strongly pulled towards the bulk of the liquid. So, hydrogen bonds are responsible for creating the cohesiveness that pulls water molecules close together, leading to a surface tension that characterizes water. It is this surface tension that allows water to form drops and allows a needle or an insect to gently or temporarily float on water. But overall, hydrogen bonds are weaker than ionic or covalent bonds. But since most of our body is water and solutes are dissolved in aqueous intracellular and extracellular solutions, hydrogen bonding greatly contributes to the stabilization of our small and large molecules, specifically proteins and nucleic acids. So on the left there, that's a picture of oxygen water molecules. And then to the right is when urea is dissolved in water. So those dashed lines represent the hydrogen bonds. They're weak, but they are formed by the attraction between the partially charged molecules. This creates a surface tension where those molecules want to kind of stay bonded together. But water makes hydrogen bonds with itself and with the other polar molecules, and that's what's shown to you on the right. So urea on the right there is a molecule produced by the liver from ammonia, and urea is polar, containing oxygen and hydrogen atoms that form hydrogen bonds with the water molecules. Since the interaction between water and urea are stronger than the intermolecular attractions among the molecules of urea, the formation of hydrogen bonds allow urea to be easily dissolved in water. So soluble urea can be readily excreted by the kidneys, which is a very effective way that we can remove ammonia from our body. It's also important because hydrogen bonds also work on large molecules such as the double helix of DNA, and will later become into play of how we have other principles in further chapters. Remember we said water really breaks down all of the bonds, so it also breaks down ionic compounds. So acids and bases and ions will completely dissociate in aqueous solutions typically, and that will release anions and cations, which in turn interact with the water molecules to maximize the electrostatic interactions and that surface tension. Water molecules will rearrange to form a hydration shell surrounding ions according to their polarity. Hydration shells also develop surrounding large charged molecules like proteins in our plasma. So when you take this all together, the properties of water not only make it an exceptional solvent, but it also works as the major transport medium in the body because it's very stable and it can carry many nutrients, gases, electrolytes, hormones, and waste. So water works very well as the transport for all of our atoms in our body, and it works very well that way. So that becomes really important to understand when we start talking about whether something readily and easily dissolves, right? Remember we said like strong acids readily dissolve, but weak acids don't. So how well an ion or an element or whatever can dissolve in water is really important because water is our main solution within our body that helps carry all of our atoms within our body. So this moves into the idea of water-loving or water-hating ions and molecules. Okay, so let's back up a second and talk about this. Page 18. Polar and ionic compounds that readily dissolve in water are hydrophilic, which means water-loving. So we want water-loving things in our body, right? We're going to be made up of 60 to 70% of water. The better these things work in water, the better off we are. So our water-loving, our hydrophilic carbs, amino acids, nucleic acids are all polar compounds and are hydrophilic. So if we remember that water is H2O, this statement makes sense. Biomolecules that have bonds between electropositive hydrogen and electronegative oxygen or nitrogen generally have polar qualities, and those tend to work pretty well in water. But nonpolar molecules are unable to interact through hydrogen bonding or that ionic interaction. So now we consider those to be water hating. They don't do as well and they're hydrophobic. So nonpolar hydrophobic molecules tend to aggregate with these other nonpolar molecules, minimizing exposure to water. And so nonpolar compounds are called hydrophobic or water-fearing. And the interactions that hold these nonpolar molecules together are called hydrophobic interactions. So if you need an example of this, think of water and oil. Okay, hydrophobic is water and oil. Hydrocarbon chains like triglycerides and lipids and fats, which is our major energy storage, are the typical examples of hydrophobic molecules, and they require special packaging in order to be transported in water solutions such as the blood. So remember our previous osmosis video specifically said fat doesn't store water, right? It's hydrophobic. It doesn't want water. It doesn't work well with it. So in water, lipids don't dissolve, right? The oil doesn't break down. You can separate the oil from the water. It's hydrophobic. The hydrophobic interactions are really important when you're talking about those three-dimensional structures that we were talking about before. So an example of this is phospholipids. They form a biological membrane within the body, but phospholipids are amphipathic molecules. Okay, you don't have to be tested on that per se. But when phospholipids are placed in water, they rearrange to hide their hydrophobic chains away from the water while exposing the polar heads towards the solution. And that extracellular space outside and the intracellular space inside. So while the hydrophobic chains are facing toward the inside of the membrane. Okay, we're going to talk about this more in chapter 4. But in essence, what we need to understand is that phospholipids do not do well in water, and they will form an aggregate together, and they kind of change the shape of themselves and kind of, I look at it as almost like bend their insides out to adjust to being in the water. Also something to consider to remember too, I'll skip back up here, is that some of the gases we have, like oxygen and CO2, they are non-polar, so they tend to be water-hating, and they are poorly soluble in aqueous solutions or in water solutions. So our body has a workaround. We have dedicated systems to facilitate oxygen and CO2 transport. So in the blood, oxygen is transported because it's bound to the protein hemoglobin and myoglobin. And then carbon dioxide combines with water and is transported as carbonic acid and bicarb, which are both hydrophilic and thus soluble in aqueous solutions. So the body has a workaround. All right, moving on to these other great organic chemistry foundations. So we're kind of on to the next new thread. I hated organic chemistry the first time, so hang in with me on this go around too, okay? Take a break if you need to, stretch your legs. We're going to move into now this kind of organic chemistry section. Carbon-based compounds are the focus of organic chemistry, and they're the principal building blocks of our body. So because the chemical properties of carbon is the leading to the formation of our molecular chains and rings of all of our organic compounds, okay, we have to understand the chemical properties of carbon. The more than 12 million compounds on which all living organisms are based are predominantly made up of carbon. hydrogen, oxygen, and nitrogen. Remember, we said we had our big main 4 molecules. So carbon has 4 electrons in the valence shell and thus is able to make combinations of 1, 2, or 3 covalent bonds. Organic molecules can contain polar and nonpolar functional groups, so hydrophobic and hydrophilic. That will determine their solubility in water. Some of the specific arrangements that occur repeatedly in the body are named and described in detail in this section. So core structures that we have, at the end of the day, the core structures that we have in our body are based on the carbon's ability to make up to 4 bonds, allowing us to have this complex organic system in our body. Organic groups that include oxygen and nitrogen can modify carbon chains and rings, creating molecules with unique functional properties. Many organic groups are polar and have characteristics of acids and bases. The addition of non-organic group to an existing molecule is described with the suffix ation. So when you have an organic group and you combine it to something else that's existing, you add ation. So methylation, phosphorylation, hydroxylation. When you remove that organic group from an existing molecule, then we have a deation. So demethylation, deamination, decarboxylation. And many reactions are carried out by enzymes with the suffix ase, carboxylase, hydroxylase, etc. So those are kind of the terms that we need to understand when we're talking about some of these organic chemistry. All right. Organic compounds and functional groups. There are 6 molecules that contain common functional groups. And there's a methyl group that consists of carbon bound to 3 hydrogens. The 4th binding site of the carbon is bound to a larger molecule. So we're on figure 2.12 in your book here. Molecules that contain common functional groups, okay? First is this one here, the amino acid alanine. This shows the methyl group in yellow, the amino group in blue, and the carboxylic group in red. This is a ribose molecule, like sugar, showing the aldehyde group in green and the hydroxyl group in purple. This is a sugar ribulose molecule showing the ketone highlighted in blue. This one here is the amino acid cysteine, which with its thiol group shown in orange. And then this is the formation of disulfide bond in orange, linking the 2 cysteines. And then this one is the amino acid sequence of insulin, showing the disulfide bonds that holds the 2 chains of the proteins together. So those are the examples that they give you in the book. A hydroxyl group consists of attached oxygen bound to hydrogen. So this one here is attached oxygen bound to hydrogen. The addition of 1 or more hydroxyl groups makes a larger structure more polar and hydrophilic. So that's what we want. A carbonyl group is a functional group in which a carbon atom is double bonded to an oxygen atom. Molecules that contain a carbonyl group are called aldehydes or ketones, depending on the location of the group in the molecule. This one is a carboxyl group. The carbon is double bonded to 1 oxygen and has a single bond to the oxygen of the hydroxyl group. This gets really confusing I know you're probably listening to this going, you're completely losing me, Sarah. Really what we want to understand is that how these groups come together is important, that they come together for a purpose when they're trying to achieve either a carboxylic group, etc. Okay. For example, another thing to understand is a group that contains amine or amino. This is a description of a group that would contain nitrogen. So that's something specific to that. The thiol group is important to understand because this has a sulfur atom bound to a hydrogen atom, right? The presence of a thiol group on the amino acid cysteine gives the molecule the ability to form certain bonds by losing the atom, okay? Don't get too caught up in all of these. But if you're a visual kind of person and this helps you understand what they're talking about, this is great. But let's kind of move on down the chapter, and I think it'll make more sense as we cover more material. So at the end of the day, our biochemical reactions in our body are trying to achieve three things. And so they will fall into a group of one of these. We either have a biochemical reaction to have synthesis, we have a biochemical reaction for decomposition, or we have a biochemical action of exchange. So when it's synthesis, this means the binding of molecules called monomers, I call it monomers, to form a complex structure, right? So we're synthesizing and we're building. So synthesis means we're binding molecules or our monomers to form a more complex structure. And many synthesis building reactions are anabolic, as they occur in the context of building up fuel stores after a meal. So in the body, glucose is stored in the liver. This is an example. And muscle in its polymeric form called glycogen. So the reactions that combine glucose molecules to form the glycogen are synthesis reactions, and they will take place in the fed state when glucose is available. Okay? So say that one more time. When we are having these biochemical reactions, where we're having the molecules that get into those different functional groups, where they're making an amine or a thiol or whatever, why do we do that? It's to serve three different types of purposes. One is synthesis, which is building, creating. Two is decomposing. And then three is exchanging or changing different actions, okay? So the three purposes of our major chemical reactions fall into one of those three. When we talk about synthesis reactions, we're putting things together to build or to make. That means the binding of molecules, which we call monomers, come together to form more complex structures. So many synthesis reactions are anabolic, as they occur in the context of building up fuel stores after a meal. Not all, but many synthesis reactions happen after a meal, for example. The example they talk about is in the body, the glucose is stored in the liver, and muscle in its polymeric form called glycogen. So after a meal, when you are in a fed state and glucose is available, you will have a synthesis reaction that's going to combine the glucose molecules to form glycogen. Okay. On the other hand, we can also have a decomposition reaction. And that takes place when a single compound is now broken down into two or more components. Most decomposition reactions are catabolic, so they're in a fasting state without energy. Not all, but most. When the glucose availability is decreased and the body performs decomposition reactions to break down the glycogen and release glucose, right? So that makes sense. When I'm going to have a biochemical reaction, when I'm combining my atoms and my anions and I'm making bonds and I'm doing these things, I could do it for the purpose of synthesizing or creating. Oftentimes, that does it in an anabolic state where we are being fed. So after we've fed our body of the nutrients and whatever we need, then is a good time for synthesis reactions. And that's, for example, when the glucose that's stored in our liver and muscle is going to be made and we're going to form glycogen. Okay? Because glucose is available, we're now going to convert it into glycogen in the liver and the muscle. But on the other hand, we can have biochemical reactions of decomposition. And that would be important because now if I'm in a hunger state, glucose is not available, I need to have a reaction to decompose and to break apart. And the example of that would be, I now need to take glycogen and go the opposite direction and break it down so I can have glucose released for energy. So that's what we're seeing here. Glucose by glycogen, when it's a synthesis, we're combining and we're forming glycogen. During decomposition, I need to take the glycogen and break it back down into glucose and glycogen. In this one here, this is showing exchange reaction entailing glucose phosphorylation. And this is that changing things. When we have an ation, means we're doing something, right? So this is a chemical reaction involving glucose that requires that it be phosphorylated by a kinase enzyme. And this exchange reaction with ATP giving up one phosphate that is transferred to glucose by hexokinase or glucokinase. Exchange reactions work when the transfer of an atom or group between two molecules occur. So what this one also talked about in your book is phosphorylation. And phosphorylation change reactions are typical examples of exchanges that entail the transfer of a phosphate from a donor, often ATP, to an acceptor molecule that could be a protein or a carb. Phosphorylation reactions have a plethora of different functions in the body. So another example is when a glucose enters a cell, it is immediately phosphorylated. So now we have a biochemical change or exchange going on with an ation, the suffix ation. So when glucose enters the cell, it is immediately phosphorylated, giving rise to glucose phosphate in an exchange reaction in which our ATP is converted into ADP, which is what we're seeing here, which is adenosine diphosphate. It's this reaction that allows cells to accumulate glucose in the cytoplasm because the phosphorylated glucose is unable to leave the cell. The release of the phosphoryl group from ATP is also used to provide energy to propel reactions that would otherwise be energetically unfavorable. Okay. So they also show here that the release of the phosphoryl group from ATP is used to provide energy to propel reactions that would otherwise be energetically unfavorable. This example is the splitting of ATP by the transport protein sodium potassium ATPase, which moves sodium against its concentration gradient from inside the cell to outside the cell, and moves potassium against its concentration gradient from outside the cell to the inside of the cell. So it's pumps like this that are going to ultimately contribute to the electrolyte distribution in our cellular compartments and will generate a membrane potential. So phosphorylation of specific protein residues is often used to regulate protein function. Okay. So again, we're kind of recapping how biochemical reactions occur in the body. Next concept they want us to understand is that biochemicals build from small to large. So we have those four main classes of our biological molecules are carbohydrates, lipids, proteins, and nucleic acids. These are also called metabolites as they are involved in the metabolic processes that take place in cells and tissue, both inside and outside of organisms. The large-scale study of all these molecules within a certain biological compartment is called metabolomics. I love that word, which is a new field of research. Since these compounds comprise an enormous structural and functional diversity, this research often focuses on specific subsets, such, for instance, proteomics or lipidomics studies, etc. With that being said, we jump into carbohydrates. So remember, carbohydrates with proteins, lipids, and nucleic acids are the four major classes of our biological macromolecules. The monomers of carbohydrates are called monosaccharides and are classified based on the number of carbon atoms they contain. So they could have 3, 4, 5, 6, or 7 carbon atoms respectively. Hexoses are the most common sugars, which is 6, which is an example to be is like glucose. So I'm going to kind of pause here. We're going to go back to osmosis and listen to this video and then jump back into our lecture.
Includes cow milk as well as human breast milk.
Let's back that up a second, Sarah. There we go. Carbohydrates. Remember, this is a review.
Simple sugars, which are little ring-shaped molecules made of carbon, hydrogen, and oxygen, either alone or in pairs, as well as more complex carbohydrates, which are formed when these rings link up together to make long chains. Carbohydrates provide us with calories, or energy, and simple sugars in particular play a lot of roles in our diet. They sweeten lemonade, balance out an acidic miso soup, fuel yeast in rising dough and alcohol, and help preserve jams and jellies. Now, sugars are found naturally in plants like fruits, vegetables, and grains, as well as animal products like milk and cheese. Added sugars are the sugars that get added to foods, like cereals, ketchup, energy bars, and even salad dressings. To be clear, even if the sugar being added comes from a natural source like sugar cane or honey, it's still considered an added sugar. In fact, a variety of ingredients listed on food labels might be sources of added sugars, some of which you're probably familiar with. Sugar actually refers to a family of molecules called saccharides. Monosaccharides, where mono means one, so one sugar molecule. Disaccharides, where di means two, so two sugar molecules linked together. Oligosaccharides, where oligo means a few, so it's 3 to 9 sugar molecules linked together. And polysaccharides, where poly means many, so it's 10 or more sugar molecules linked together. Glucose is the most important member of the sugar family, and it's a monosaccharide. It's one of the main sources of calories for the body, and is able to cross the blood-brain barrier and nourish the brain. Another monosaccharide is fructose, which is commonly found in honey, fruits, and root vegetables. Finally, there's the monosaccharide galactose, known as milk sugar. It's known as milk sugar because it's only found in nature when it links with glucose to form lactose, a disaccharide found in the milk of mammals, which includes cow milk as well as human breast milk. Sucrose, or table sugar, is another disaccharide, and it's formed when fructose links up with glucose. Sucrose is found in various fruits and vegetables, with sugar cane and sugar beets having the highest quantities. Maltose is another disaccharide, and this one is 2 glucose molecules linked together, and it's found in molasses, which can be used as a substrate to ferment beer. Sugars, like fructose for example, are most always found in combination with other sugars, and the combinations can be pretty different, even in seemingly similar foods. For example, in honey, 50% of the sugar is fructose, 44% is glucose, 4% is galactose, and 2% is maltose. Whereas in maple syrup, less than 1% of the sugar is fructose, 3% is glucose, and 96% is sucrose. So simple sugars, whether they're natural or added, are mixtures of monosaccharides or disaccharides. Next, there are the complex carbohydrates. There are oligosaccharides, like galacto-oligosaccharides, which are short chains of galactose molecules, like those found in soybeans. Then there are polysaccharides, which are even larger chains with branches, and are the most abundant type of carbohydrates found in food. Starches are polysaccharides with molecular bonds between sugar molecules that human intestinal enzymes can break down. Starches are an important source of calories and can be found in foods like rice, potatoes, wheat, and maize. Starches don't taste sweet like simple sugars because they don't activate the taste buds in the same way. And there are also dietary fibers, which are carbohydrates that intestinal enzymes can't break down, and so the body can't digest them. Now, there are many different types of dietary fibers, and they're not all the same when it comes to their structure or impact on health. Fibers have molecular bonds that are resistant to human intestinal enzymes, so they pass through the small intestine undigested, get broken down a bit by bacteria in the large intestine, and ultimately end up as bulk matter in the stool. Fibers are critical because they can slow down the rate of absorption of simple sugars like glucose in the small intestine, which can help maintain healthy blood glucose levels. They also increase stool weight, which helps prevent constipation. And fibers like beta-glucan are also good for heart health. Monosaccharides link together through glycosidic bonding, which is when an OH group from the carbon on one monosaccharide binds with the hydrogen from the carbon of another monosaccharide. Together this forms an H2O, or a water molecule, which goes away. In the case of maltose, that leaves an alpha-1,4 glycosidic bond, which is a bond between carbon number 1 of one glucose monosaccharide and carbon number 4 of the other glucose monosaccharide.
Not testable. We are just reviewing generally here of how these processes work.
And alpha refers to the fact that the molecules are lined up next to each other. Lactose, on the other hand, has a beta-1,4 glycosidic bond, meaning that carbon 1 of galactose and carbon 4 of glucose are bonded, but this time the molecules are stacked with one higher than the other.
This also is what we were talking about with the molecular geometry. Like the shape of it is different, but depending on the bonding. So this is a good illustration of that kind of geometry we were talking about earlier.
Finally, sucrose has an alpha-1,2 glycosidic bond, meaning that carbon 1 of glucose and carbon 2 of fructose are bonded. Now, when you eat something, like a piece of onion bread, enzymes start breaking down the disaccharides, oligosaccharides, and polysaccharides into monosaccharides so they can be absorbed. Different enzymes help to break down different linkages. For example, amylases break down large polysaccharides like starch into smaller units, whereas lactase, sucrase, and maltase break down lactose, sucrose, and maltose.
You can see that these are all ases, like we talked about. So this is part of the process of the biochemical deconstruction or decomposition
Into their monosaccharides. The individual monosaccharides that result from the digestion of larger carbohydrate molecules, glucose, fructose, and galactose, cross the gut lining and get into the bloodstream to get used by the body. When glucose levels in the blood increase after eating, the pancreas releases the hormone insulin, and this helps move glucose into all cells for energy. Insulin helps stimulate the liver to store glucose as glycogen in a process called glycogenesis, which is when some of the glucose molecules get linked together with alpha-1,4 and alpha-1,6 glycosidic bonds to form a polysaccharide called glycogen. Insulin also promotes fat and protein synthesis. Now, metabolism of galactose has an initial step where an enzyme in the liver converts galactose into glucose. It basically flips the orientation of the OH group on the fourth carbon. Just like that, galactose can become just another glucose molecule in the liver. Fructose, though, is handled a bit differently by the liver. Fructose has a total of 6 carbons, and most of it's broken down into 2 3-carbon molecules and sent into glycolysis to help generate energy.
This is not testable to memorize any of these specifics. We're just talking general terms, understanding what's happening.
When energy is needed, all 3 monosaccharides are metabolized through glycolysis, then the citric acid cycle, and oxidative phosphorylation. Ultimately, all digestible carbohydrates, regardless of whether they come from simple sugars in honey or from starches in baked potatoes, are broken down into their component monosaccharides for immediate energy use or stored away for a rainy day, depending on what the body needs. The National Academies of Sciences, Engineering, and Medicine recommended that a healthy diet contain 45 to 65% of its calories from carbohydrates. The number of calories you need to maintain your weight depends on things like your age, sex, height, weight, and activity level. For example, let's take this slightly active 40-year-old woman who's 5 foot 9 inches and 160 pounds, with a BMI of 23.6 and requires a 2,000 calorie diet. And let's say she wants to aim for 55% of her calories from carbohydrates. So that's 55% of 2,000 or 1,100 calories. And there are different types of carbohydrates. First, there's fiber. The general recommendation is to get 28 grams of fiber in a 2,000 calorie diet. With roughly 0 to 2 calories per gram of fiber, that's about 56 calories, or about 3% of her total calories. Next, there's sugars, both the kind that are added to foods and those naturally found in whole foods. There are few formal recommendations for total or natural sugar intake. However, updated Canadian nutrition labels are based on a daily value of 100 grams or 400 calories from total sugars, which is 20% of a 2,000 calorie diet. When it comes to added sugars, both the World Health Organization and the US Dietary Guidelines recommend that they make up fewer than 10% of total calories. Just like her goal for total carbohydrates, she's aiming for the mid-range of the added sugars recommendation, which would be 100 calories or 5% of her total calories. Using this approach, the remainder of her total sugar calories, 300 calories or 75 grams, would come from the sugars found in fruits, vegetables, dairy, and grains. This would be 15% of her total calories, and that leaves 640 calories or 160 grams from starches, or 32% of her total calories, to reach her carbohydrate intake goal. Now, eating a healthy diet means choosing foods that are as nutrient-rich as possible, and foods that contain fiber, starch, and natural sugars like fruits and vegetables tend to be richer in nutrients than those with added sugars. Having said that, processed and packaged foods are a part of most people's diets, so carefully reading nutrition labels can help you compare foods and choose more nutrient-rich options. Generally speaking, picking foods and beverages that are higher in nutrients like fiber and lower in added sugars is best. Alright, as a quick recap, there are various types of carbohydrates. Simple sugars are monosaccharides and disaccharides that the body can readily absorb. Starches are polysaccharides that take longer to absorb. And fibers are polysaccharides that the body can only partially absorb with the help of gut bacteria. Ultimately, a healthy diet includes all types of carbohydrates, coming from a variety of sources like fruits, vegetables, dairy, and grains. It can include added sugars too, with the World Health Organization and the US Dietary Guidelines recommending that added sugars make up less than 10% of your overall calories.
So jumping back into the carbohydrates from our book, we have a little more definitions we have to kind of talk about to understand here. Carbohydrates contain chiral carbons, which are carbon atoms bound to 4 different substituents. So they can exist in 2 different forms called enantiomers, which is D and L forms of carbohydrates, have the chiral carbon oriented in such a way that one enantiomer is the mirror image of the other. So we're talking about what the structures look like. The D forms of carbohydrates are the most prevalent. A hexose may be present in its linear form or may cyclize, leading to the development of a hexagon or pentagon structure, which is what they talk about here. Common forms of the hexoses include galactose and fructose, as well as pentoses, ribose, and deoxyribose. Okay, so D and L are the most common in nature. Ribose is a 5-carbon, and deoxyribose we talked about. The disaccharides consist of 2 monosaccharides binded together. So sucrose was the glucose and fructose. Lactose is glucose and galactose. And maltose is glucose plus glucose, many starches. I do want you guys to have an understanding of that concept there. The hydroxyl groups on the sugars make them very hydrophilic, so water-loving. They are easily soluble in water. They cannot diffuse across cell membrane lipid barriers, so they require carrier proteins. D and L are the most common, D and L forms, but D is the most common. They are our principal source of energy, and they're the sole energy source for the brain under most conditions, which is glucose. The liver regulates blood glucose by synthesis of glycogen after meals, release of glucose from the glycogen while fasting. Remember the synthesis or the decomposition. Our optimal levels of glucose is 70 to 110. Hyperglycemia occurs in diabetes and leads to chronic problems. And then hypoglycemia can cause the brain dysfunction and coma. So we had D and L forms. D is the most common. This is the example of D-glucose. I also want to point out that we talked about up here that lactose is glucose plus galactose. Absence of the enzyme lactase leads to lactose intolerance. Because lactose is made up of the glucose and galactose, so that when you digest milk and dairy products, that lactose, you have that enzyme lactase that's broken down into its monomers, which are then assimilated. Also, when we talk about them being very hydrophilic, that's a really good thing, because that means in water, these groups can easily form hydrogen bonds with the water molecules, and because such polar interactions are greater than the interactions among carbohydrate molecules, they readily will dissolve. This hydrophilicity of carbohydrates is a great advantage because it allows them to be easily solubilized and therefore transported in water, including our plasma and cytoplasm. But carbohydrates are poorly soluble in hydrophobic environments such as membranes. So for that reason, remember, carbohydrates are unable to cross the hydrophobic portion of the biological membranes, and they will require carrier proteins. So again, going back to glucose, I'm sorry, I jumped. Almost all the cells in our body, again, will use glucose, so it is the principal source of energy. Remember that the brain needs it a lot. It needs constant glucose levels to be steady in the brain. Carbohydrates are also really important because we use them to modify the components of the cell membranes. Proteins modified by the addition of a carb are termed glycoproteins. Lipids modified by the addition of a carbohydrate are termed glycolipids. Glycoproteins and glycolipids are abundant in our plasma membrane, and that forms a glycocalyx, which is a sugary membrane coat. And those molecules play diverse roles in the body, okay, because they can provide support or attachment sites, they help transport other molecules, they can be signals to hormones, etc. So carbohydrates, and especially when it breaks down into D-glucose, is an incredibly important source of energy for the brain, and it really can help modify cell membranes to be used in other processes. One of the examples your book gives, which I think is very interesting, is our ABO blood groups are an example of the membrane-bound carbs responsible for distinguishing self from non-self. Thus, they're an important part of immune function. So the groups are determined by the presence of carbohydrate residues that are found on the surface membrane of the red blood cells in the form of a glycoprotein or a glycolipid. The sequence of the oligosaccharides determine whether the antigen then is A, B, or O. So that's where we get this. This shows sphingolipids bound to A, B, or O groups. Individuals with the blood type A have a modified form of galactose, N-acetylgalactosamine, at the end of the glycolipid. But those with type B express galactose instead. Individuals with the blood type AB express both types of the glycolipids on their red blood cells, and those with the blood type O express neither galactose nor N-acetylgalactosamine at the end of the glycolipid or glycoprotein. So transfusion reactions will occur, and they can be fatal when blood is not correctly matched between the donor and recipient. So carbohydrates are essential in our ABO part of our blood, which is really cool, in my humble opinion. We are going to end part 1 here at the end of carbohydrates, and I will see you in part 2, and we'll wrap up the rest of the chapter.